Hello, everyone. Thank you for attending the 2024 Hugh Davson Distinguished Lectureship Award presentation of the APS Cell and Molecular Physiology section, titled Calcium, ATP, and ROS, a Mitochondrial Love-Hate Triangle. My name is Dr. Amity Eaton. I'm a postdoc at Mass General Hospital, and I'm here with my co-moderator, Dr. Jin O. Uchi from the University of Minnesota. And on behalf of APS, we would like to thank everyone for attending today's webinar. Before we begin, I would like to present a few housekeeping notes. First, the webinar is being recorded and will be available for on-demand viewing. Second, to maintain scientific integrity and respect towards our distinguished lecturer, please refrain from photo or video capture of this scientific presentation in whole or part. And finally, during the lecture, please post your questions and comment via the Q&A feature instead of the chat. You may post them at any time during the lecture, and they will be read when the lecture concludes. So next slide, please. So the Davson Award Lectureship is the top honor bestowed by the Cell and Molecular Physiology section of APS, recognizing a scientist who has made major and meritorious contributions to the scientific areas represented by the section. The award is named for Dr. Hugh Davson, who was well regarded for his work on ion transport in ocular fluids. So without further delay, I'm honored to introduce our lecturer today, Dr. Shih-Ching Hsu. Next slide, please. So Dr. Hsu is the William Wyckoff Smith Endowed Chair and Professor of Cardiovascular Research at the Center for Translational Medicine at Thomas Jefferson University. His research focuses on the role of calcium, ATP, ROS, and mitochondrial fission-fusion dynamics in regulating heart function under both physiological and pathological conditions. He has published close to 150 papers, which have been cited over 16,000 times, and many of them in APS journals. He has trained almost 40 graduate students and postdoctoral fellows, most of whom continue to conduct research. Even after running his lab for 41 years, Dr. Hsu is still passionate about his research, which motivates him to wake up at 4 a.m. every day to catch the first train to his lab in Philadelphia. Luckily for us, we didn't have to get up at 4 a.m. to hear his great talk. So congratulations, Dr. Hsu, and now I'll turn it over to you for your presentation. Okay, looks great, Dr. Hsu.
Thank you. Okay, so looks good. Thank you so much for the introduction, Dr. Eaton. And before I start, I really want to thank the supporting staff from the American Physiological Society, particularly Patricia Terrell. She really guided us through this very adventurous event, and thank you so much. And of course, Dr. Eaton and Dr. Uchi and Dr. Sandrine Pierce from the APS. So this is the title of my talk. And before going into my talk, one of the reasons I wake up so early in the morning is I grew up in a very small town in Taiwan. And by the time I went to the National Taiwan University, the Department of Physics, all of a sudden I realized there's so many smart people around me. And so in order to catch up with them, I just had to work harder. And let's carry on since then. And at this moment, I still surrounded by so many smart people, and therefore I keep working harder and harder, even as of today. Okay, but first of all, I want to thank the American Journal of Physiology. As Dr. Eaton mentioned, I published 14 papers, which is about 10% of my publication, but has been cited over 4,500 times, which is over a quarter of my citations. So thank you, and it really helped in terms of my scientific careers. And I'm going to talk about the triangle, the calcium, ATP, and the ROS. And this paper has been cited over 3,000 times. And as such, I feel this is an important topic. Why it is an important topic? Because this triangle control the flow of the vital force and energy through our body. And in Chinese, we call it qi. And thus, basically sustain the life, health of the human being. So this is a very important question. And let me use this slide that I adapted from Doc Wallace's review article. When you think about all diseases, either they are coming from the nuclear DNA mutation or epigenomics, or the mitochondrial DNA mutation, or from the environment factors, they all converged into three things: energy, ATP, ROS, and the calcium. And from there, basically, you can explain almost all kinds of disease. Either it is neuro, psychological diseases, cardiomyopathy, cancer, aging, metabolic disease. It's all dwell upon the balance of these three factors. And my research focused on the cardiac bioenergetics. Why this is an important question? Because each day our heart going to use 6 kilograms of ATP for the pumping of the blood. So if our heart doesn't, the mitochondria in the heart doesn't regenerate the ATP, then in 1 minute, all the ATP in the heart cell are going to be completely used, and therefore the life will cease. So there's a very important fundamental question about this phenomena, namely, how can the cardiac muscle cell make 4 grams per minute of ATP, which is about the weight of a nickel? This is a fundamental question, which even as of today, we all thought we already have a textbook answer to these questions. And the question, of course, is related to the calcium, okay? The idea is calcium, during the excitation, contraction, coupling of the heart, a small amount of the calcium goes through the L-type calcium channel that causes the calcium-induced calcium release. From the SR, that calcium release contribute mostly to the muscle contraction, the pumping of the blood. But there's a very small amount of the calcium go inside the mitochondria and regulate 3 key enzymes in the TCA cycle. As such, on the B2B basis, it can replenish the substrate for the electron transport chain and produce the ATP. So this is a beautiful textbook principle in terms of what we call excitation, contraction, metabolism coupling. So in order for this to occur, then the mitochondria has take up calcium very rapidly. Remember, our heart rate is about 70 beats per minute, and then the mouse is about 550 beats per minute. So how can the mitochondria in humans or human heart or in the mouse heart take up the calcium so quickly? And there are two reasons for that. One is there's a privileged calcium transport, which I will show you a little bit of data, that transport directly from SR to the mitochondria due to the physical contact between these two organelles. And the second one is our more recent publication about this biased distribution of mitochondrial calcium reporter and the sodium calcium exchange. Okay. And this is a paper we published with Clara Franzini Armstrong. In this case, it's the EM picture of this is a T-tubule, which is extracellular space. And this is the sarcoplasma reticulum. And Clara said these are the feet, namely the ryanodine receptor. But for us, the most exciting aspect of this EM image is mitochondria are in contact with the sarcoplasma reticulum in the adult cardiomyocyte. And this contact create what we call the nanodomain or high calcium during the calcium release from SR to the mitochondria, okay, or SR release for the muscle contraction. And let me use this Irvin Nail, the Nobel Prize winner, figure to explain what I said, the nanodomain. Let's think this as a ryanodine receptor. And let's think this as the mitochondrial calcium uniporter, okay? If they two are within the nanometer distance, then opening of one calcium channel, ryanodine receptor, in this nanodomain, there will be a 100 micromolar increase of calcium. Remember that cytosolic calcium is about 100 nanomolar. And this will go up and down in terms of microsecond. And if you put some kind of high calcium chelator, you cannot buffer this because it's so quick and it's so high. And from this point, I actually want to remind you, this makes the measurements of calcium pulses during excitation contraction coupling, say, go into mitochondria, extremely difficult because of high concentration as well as very rapid rise and falls, okay? But this is the concept of microdomain of calcium. Does it happen in the adult cardiomyocyte? So we did a simple experiment. We take one single cardiomyocyte and we use chemical skin the plasma membrane. As such, we can control the what we call quote unquote cytosolic environments. And we use the caffeine to dump out the calcium from SR and measure, in this case, cytosolic calcium. And you can see a robust increase of cytosolic calcium. And we use a very high affinity calcium chelator beta, and put the caffeine, then the caffeine pulses in the cytosol, mostly buffered by the peptide, okay? But now this is the nanodomain concept. Now we measure mitochondrial calcium and we dump the caffeine, and we see a huge calcium increase, but the beta, high concentration beta, millimolar, no longer can chelate the calcium that go from SR to the mitochondria, just like the figure that Irvin Nail showed. There's a privileged transport of calcium from SR to the mitochondria in cardiomyocytes. So at this point, I also want to say, measuring this many studies attempt to measure mitochondrial calcium during the heartbeat, okay? And the published data, not surprisingly, it ranges from each beat can be 10 nanomolar or up to 10 micromolar. And this mostly due to the reason that I mentioned before, either the probe doesn't have the affinity or the probe doesn't have the on and off binding constant that can rapidly measure the calcium transient. And that's why the variation is enormous in the literature. And the other aspect of the mitochondrial calcium uptake currently, the belief is mitochondria do not take any calcium at the cytosolic or physiological calcium concentration because the MIQ1 is the gating mechanism that stop mitochondria from taking up calcium until the calcium go up to micromolar range. So the dogma is mitochondria physiologically do not do mostly anything. Yeah, but I also want to share with you one of our data that actually show mitochondria can buffer calcium under physiological condition. In this case, we isolate mitochondria from rat heart, purify mitochondria from rat heart, and we inject these cardiac mitochondria from rat heart into Juniper's oocyte, okay? And this is a control without injection on the cardiac mitochondria. And if we put a fibrogargan to release the calcium from ER in the Juniper's oocyte, you can see a calcium pulses. Beautiful. But after injection of the cardiac mitochondria into the Juniper's oocyte, look, this calcium pulse is completely blunted and it becomes slower. Suggestion mitochondria are buffering the calcium in the cytosol. And indeed, we also measure the uptake of mitochondria. Look at the cytosolic calcium is less than 100 nanomolar. And the mitochondria can tap our calcium under physiological state. And I think this is an important point. That's why I want to bring this up. Even as today, this is still a very controversial issue, okay? And the second part, now I address the nanodomain. The second part is what I call the bias distribution of the mitochondrial calcium transport system. And the result we have shown is most of the mitochondrial influx system, MCU, are localized nearby the SR contact site. And most of the sodium calcium exchange is away from that contact site, okay? And as such, it will create what we call a calcium gradient within one single mitochondria. And this brings us some of the concepts that we are working on in terms of zoning or the mitochondria. Maybe within one single mitochondria, there's two different zones. One zone is for the calcium uptake, the other zone is for the energetics. And this is basically a cartoon to show you physiologically this makes sense. L-type calcium channel release the calcium from SR, MCU nearby, take out the calcium, and then go to the into the mitochondria, and subsequently pumped up by the sodium calcium exchange. And then where the network SR are located, they can reuptake the calcium for the next calcium, the beat. And therefore, the cell are happy, okay? I think I go a little bit too quickly. So everything is fine. But if the sodium calcium exchange and the mitochondria are locally around the same area, because sodium calcium exchange is electrogenic, and that will cause this depolarization of mitochondrial membrane potential, then that would defeat the purpose of the energetic, which require the ATP membrane potential. And therefore, it makes sense they distribute with a preferential location. Okay. So the next question I want to address to you is about, okay, so MCU is the one that bring the calcium in, and lastly in the textbook, yeah, that bring the calcium into mitochondria and turn on TCI cycle and make the energy, okay? And so the question is the MCU the one and only mitochondrial calcium influx mechanism? And that's what the research community did. Why did he embrace the idea? MCU is the only calcium influx mechanism in the mitochondria. Okay, and about 13 years ago, finally, after so many years, these two beautiful papers show that we finally molecularly identified the MCU as such open up the field, because now you can use a genetic tool to knock in the MCU in heart, in kidney, in liver, in brain, and so, and that is ongoing research in many, many places. But surprisingly, in the heart, either use cardiac-specific MCU knockout, or you use the global MCU knockout, there's no phenotype. This is very, in a way, very disappointing, okay? As I say in the first two slides, in 1 minute, if the mitochondria do not regenerate, the heart going to die because the energy is completely depleted. So what's going on? And this is not just the heart. And I think if you Rudo, who identified in a nature paper, they use the neonatal, so they can use the siRNA knockdown and see they know energetic phenotype. Venzi Muta is another one who that side-by-side nature paper, and he found in the brown adipocyte tissue, knocking out MCU has no energetic, and so on and so forth. So this is a very puzzling observation. And so for me, then I start to ask questions. If the MCU is dispensable for the excitation bioenergetic coupling in the heart, then what are the other alternatives? And we and some other people, maybe there's more than just one calcium influx system. Indeed, the MCU may account for 80–90%, depends on the range of calcium we are talking about. Is all calcium dependent and the time dependent, so on. So maybe there are other that maybe not as robust, but they can sense the take the calcium, like latent 1, TRPS3, UCP, and our discovery of type 1 mitochondrial ryanodine receptor, as well as the rapid mode of mitochondrial uptake. Okay, that's one possibility. The second possibility, maybe it's still calcium. And maybe it's still calcium in the extra mitochondrial nanodomain. But it's like calcium outside of mitochondria is doing something to send a signal for the excitation bioenergetic coupling. So these are two other possibilities, okay? And as I mentioned, we identify the type 1 ryanodine receptor as a rapid mode along with the MCU. And this was late Tom Gunter. We discovered this quite a long time ago, 1995 and 2001. Yeah. So that's one possibility. The second possibility, as I mentioned, does only calcium go inside the mitochondria can do the job of excitation bioenergetic coupling, okay? And we don't think so. So we start to look at the possibility, and in the next few minutes, I'm going to share with you is our work on the non-canonical role of DLP1 in excitation bioenergetic coupling. And this is mostly in collaboration with Wang from the University of Washington at Seattle. Okay. And this is again from my collaborator, late Ethan Hume. We collaborate, and I learned so much from Ethan. And he was one of the first who discovered the DLP1. And this is from a cultural liver cell. And you can see mitochondria are constantly in the cultural liver cell, go through the fusion, fission, and the trafficking. Okay. And this is a very pronounced physiological and biological implication. But look at the adult cardiomyocyte. About 35% of the volume is occupied by mitochondria for a good reason, because they need to make a lot of energy. The other, they have no place to move, because the other 2/3 of cell are the contractile proteins. So unlike the culture liver cell, they cannot go anywhere. And also, if you look from the confocal imaging and so forth, the fusion and fission event occur, relatively speaking, in adult cardiomyocyte is much, the frequency is much smaller than other cells, including the neuron. Yet, at the meantime, people find the fusion, fission protein like DLP1, mitofusion-1, mitofusion-2, OPA1 are most abundantly expressed in heart as well as neuron, okay, in the excitable cell. So what is the reason when they don't do the fusion and fission, yet they have such high level of mitochondrial dynamic proteins? And that comes to our thinking about the non-canonical role, okay? So a few years ago, with Wang poster, Hui and Zhang, we used the siRNA infection, or use the dominantly DLP1 form, which is K38A, to inhibit the DLP1. And in the adult cardiomyocyte, yes, we saw a little bit of the change of morphology. They tend to be a little bit longer based upon the aspect and the form ratio. But most surprising observation is this almost 300% decrease in the respiration, the linked to the ATP generation. We did not see much of change of morphology, but we saw a huge decrease of the mitochondrial respiration when we inhibit the DLP1. So what's going on? And let's come to our thinking. This is what we call mitochondrial flashes, which is this is loaded CPYFP. This is from my collaborator, Dr. Davson, the lab in skeletal muscle. Mitochondria are stochastically burst into three things. One, depolarization. Two, what we think is superoxide generation. Three, pumping of the proton, the pH. Okay, so these flashes, and these flashes can be blocked mostly by cyclosporin A, which is the inhibitor for the mitochondrial permeability pore transition. So what's going on? Okay. And we summarize in this slide. So mitochondria, now we have to think them as they can be like an excitable cell, okay? And at the quiescence, okay, they stay on with the membrane potential, calcium, ROS, and so on and so forth. But for some reason, we still don't know yet. There's a sudden transient opening of mitochondrial permeability pore transition. And that suddenly mitochondria depolarize. And in order for the mitochondria to bring back the membrane potential, they have to enhance the robust electron transport chain in order to bring back the membrane potential. As such, we think there's a huge increase of bursting of the ATP generation. That's number one. Number two, there's a bursting of superoxide generation because you have such a high frequency of electron flow, and some of electron going to bind to the oxygen and produce the superoxide. And thirdly, because of electron transport chain pumping, you had a proton fluxes. So you will see a sudden alkalization of the mitochondrial membrane potential. And that comes to the point of these three things, ATP, ROS, and proton. You can think that as and the permeability pore transition, which linked to the calcium triggering. And so these superoxide flashes, we think is a indicator of the energetic state of the mitochondria. And the DLP1 had this non-canonical role that somehow sent some signaling to open the permeability pore transition transiently, as such can enhance the energetic as well as ROS and the calcium. And that's why when we knock down or block the DLP1, there's a drop in the mitochondrial respiration. Okay, I had 10 minutes. I need to go quicker. So in order for that hypothesis to be validated, then we have to assume the majority of the DLP1 should be localized in the sarcoplasmic reticulum and the mitochondrial junction and talk to the MPTP, as such, talk to the energetics, okay? And because of time, I'm going to rush a little bit. And the theory indeed show the mitochondria associate DLP1 all in the MAM fraction. MAM is mitochondria associate SR membrane. And then in addition to biochemical study, this is to show you the confocal, they are very close to the ryanodine receptor, which is indication they are close to the MAM of the SR mitochondria. And this is the EM study. You can see this big gold particle. They are in a T-tubule and SR junction that contact to the mitochondria, and this is statistic. And this is what the biochemical study. So this is DLP1. The majority of DLP1 is in the cytosol, but they are also in the SR and in the crude mitochondria. And then when we purify the crude mitochondria to remove all the SR contamination in the pure mitochondria, you don't see any DLP1. All the DLP1 is in the MAM fractions, which support the idea there in the junction, okay? So to conclude this part, so we think that the potential mitochondrial excitation can there are two ways. One is still dogma, the textbook, the calcium go inside, could be MCU, could be others, that trigger TCA cycle, enhance, and the other we think is a DLP1 can do the permeability. Then there are more recent study, people start to look at that. Maybe this you can enhance the substrate like glutamate, pyruvate, and or ATP sends to the microdomain calcium and transport into the mitochondria instead of mitochondria regenerate through the TCA, and that can enhance the TCA cycle. And then, so these are all the other possibility, and I'm not going to talk about MPTP because of the time, okay? And so to summarize this part, there's a previous calcium transfer from SR to mitochondria, okay? Through the bias distribution, knockdown has very little effect on EB coupling. Although MCU dominant mitochondrial uptake, there are the calcium influx system. The high heart rate challenge the recording of B2B. I wish there would be a probe that can sense microsecond change of the calcium with a good affinity. And DRP1 may play a role in the EB coupling through the transient opener. And the fundamental mechanism, unfortunately, after today, I think it still remains an enigma. And the heart failure is an engine that runs out of fuel. Therefore, to resolve this mystery is very important for the effectively treating life-threatening diseases. How many minutes? I said 7 minutes? Okay. And because the triangle, I spend most of my time on the calcium ATP, but calcium also does the ROS generation. And this can be presented by the next 2 slides, okay? And this is from my former graduate student, Jennifer Holmes, work with Ethan. If she uses ceftriaxone to release sustainedly high calcium from the ER that's sustainedly high in the cytosol, those mitochondria that did not have the DLP1, K38, they all become fragmented. That's number one. Calcium can fragment mitochondria. That is DLP1 dependent. That's number one. And number two, this is a neonatal cardiomyocyte. This increase of effective can increase of cytosolic calcium lead to an increase of ROS, okay, as you can see in here. But that increase of ROS disappear when we inhibit the DLP1, in this case, use the K38. And the mitochondria still can make the ROS because you use the complex 3 inhibitor, they still goes up. So let's summarize this part of the study. The mechanism of mitochondrial calcium, either in the mitochondria or in the cytosol to induce ROS, is through either activation of DLP1 non-canonically or canonically through the mitochondrial fission, and that lead to the increase of ROS. And then again, there's a crosstalk between ROS and MPTP, and then it can have this cycle or the ROS, MPTP, more ROS, and so on and so forth. Am I all right with time? Okay. And again, it's triangle. The ROS can talk back to the calcium. And this is what I call the Goldilocks principle of calcium ROS crosstalk signaling. So when the mitochondria are making energy, they also make ROS. And that ROS is important for signaling. And that ROS can talk to the renin receptor and make them a little bit more effective in terms of release calcium. So everything is good, okay? But then when the calcium becomes so excessive, like in the case of Jennifer's study, that have get sustained increase, then that too much of ROS can really oxidize renin receptor, and that can make the renin receptor very leaky, and that is dangerous in terms of cardiac arrhythmia. And indeed, our collaborator from Albany, the husband and wife team, Yu Min Zhang and Yong Xiao, we start to work on this kind of concept. What they have done is they overexpress the heart with type 1 myosin receptor, and they saw an increase of ROS after the overexpression. Also, the heart actually become hypertrophy, and then that, as I say, can cause the leaky myosin receptor, which can be monitored by the calcium spot. There's more calcium spot, and this is actually the calcium spot. Also, is the way the leaky myosin is the way that people used to explain the life-threatening catecholaminergic polymorphism ventricular CPVT. Okay, then so we start to look at the mitochondria, talk back to the SR, and induce a cardiac arrhythmia as well as the heart failure. So I'm almost done. Okay, so the second summary, like calcium promote mitochondrial fusion of DLP1 mediate mitoflashes through the non-canonical role, which lead to the ROS generation. ROS in the mitochondria SR junction oxidize the nearby calcium transport protein, including the type 1 myosin receptor, and change their activity. The calcium and ROS crosstalk signaling promoted excitation, contraction, bioenergetic coupling physiologically. Okay. However, excessive mitochondrial calcium uptake lead to the overproduction of ROS. This vicious cycle of calcium and ROS overproduction cause cardiac arrhythmia and heart failure. So where am I going from here? I think my timing is all right. Yeah, okay. I have 2 more slides. Where are we going from here? Keep me waking up 4 o'clock in the morning, I guess. Yeah. One direction we want to go is, recently it has been shown the ATP synthesis, complex Y, and the ANT, they are permeability for transition for candidate protein. So each mitochondria has 100,000 copies of the ATP synthesis NT. But if you open one permeability pore transition, if it's long enough, it collapses all membrane potential. So the question is, how can among these thousands of ATP synthesis and ANT, why does 1 or 2 behave act MPTP? And we'd like to find out what's the reason, okay? And the second one, now we feel that DLP1 has more function in adult cardiomyocyte more than efficient. So we start to look into what's the signaling mechanism and why they are locular in there. So we specifically looking at actin and actin binding protein in terms of fixing the DLP1 in the mom. Okay, and then of course there's thousands of protein in the mom, and so what's the interactome and how that link to the excitation bioenergetic coupling. And lastly, we have to go beyond SR mitochondrial communication. And recently, we artificially generate mice that have artificial linker that link the SR and mitochondria with a fixed distance, say 5 nanometer or 10 nanometer, whatever. And then we look at the heart from this linker mice. The mitochondria start to cluster together and they start to form this nanoternal. They start to talk to each other through this nanoternal. And we think this is a fundamental issue about mitochondria communicate with mitochondria. And after all, mitochondria coming from bacteria, they like to form quorum, they like to communicate. Whether this nanotone has anything to do with the mitochondrial communication, which is what we like to do next. And one more, this is the last slide. Optimistically, one can say never too old to learn. Or pessimistically, you cannot teach this old dog how to new tricks. We start to work on mitochondria, what we call quantum bioenergetic. And the whole idea came from about 12 years ago from late Arthur Moss at the University of Rochester. And the author come to me and say, why pacemaker, the patient implant with pacemaker around the area of my heart cell look healthy? And I say, must be electromagnetic field. And that was 12 years ago. And now we come to the time we may understand what's the reason, and that reason may has to do the mitochondria contain lots of iron sulfur cluster and the adeноnucleotide, and a very small magnetic field can form the radical pair and change the spin state or electron, and then that may alter the energetic. And finally, I will present this sometime in August. All this work was done by the people who are working in my lab, and I don't have time to go through each one of them. I mostly just come in early in the morning. Actually, the first thing I go to the gym to play squash. Yeah, not really working. And I have a lot of collaborators, and I appreciate throughout the year all the collaboration with so many friends and colleagues. And then lastly, I want to thank you, and please feel free to contact me through this email or the website, and visit my lab in Philadelphia. You are always welcome to stop by and say hi. Yeah, thank you so much. Yeah. Stuff. Yeah.
Thank you very much, Dr. Xu. And the presentation is closed, and we would like to move to a question and answer session. So the first question that we would like to start is from, I'm trying to pronounce that correctly, but if it's not, I apologize. From Dr. M.D. Abui Hopkins. Dr. Hopkins' question is as your presentation is mostly the calcium movement from ER or SR to the mitochondria. But is there any condition that the calcium goes from mitochondria to SR, ER side? And if it's happened, what kind of condition?
Yeah, the question, of course, yes, because whatever comes in has to go back, right? It's not like cockroach motel, it goes in, it doesn't go out. Okay, now I'm giving a joke now. Okay, so as I show in the cartoon, the mitochondria, so that's why it's beautiful in the way the sodium calcium exchange away from junctional SR. And around that area, there's this network SR that wrap around the mitochondria. So the sodium calcium exchange pump out the calcium from that mitochondria, they immediately go to the network SR. So there's indeed also the transport of calcium from the mitochondria to the SR.
Great. So let's move to another one.
Okay, Sam Kongsamut asked, does DRP1 knockdown impact cardio function in vivo?
Yes, DRP1 knockdown tremendously. If you knock out DRP1 in the actually embryonic lethal, that's number one. So we generated DRP1 cardiac specific knockout mice, and the heart basically start to develop arrhythmia and the energetic issue, and they die about 3 months or maybe 2 months after the knockout. So and then mostly I think they die because of cardiac sudden death because of alteration of energetic as well as the calcium. And we are studying that now. Yeah, come to visit us. Yeah.
Great. There's several questions that similar question. One is from Dr. Paul Brooks and also from Dr. Michael Cypress regarding about the calcium secretion or the buffering inside the mitochondria, for example, including calcium phosphate granulate. So how does the calcium buffering capacity or the mechanism also impact to the signaling what you're looking?
Yeah, that's a very important question. And my point of view, there are two aspects. The buffering tend to be under the condition when there's a calcium overload, okay, like ischemic reperfusion injury. And I think calcium phosphate, the buffering, as well as other calcium binding protein, play a critical role in terms of buffer. Okay, so that's somewhat more pathological. But then I look at my data or inject mitochondria into xenopus oocyte, which is very physiological. So how do I connect that? And indeed, it play a tremendous role in the buffering. So I don't know. But the reason I'm kind of hand waving in a way is that the general scientific community still think mitochondria, quote unquote, buffering doesn't happen under more physiological condition, but it could be that way based upon the observation we have. But then the question is whether that linked to the phosphate or other things that I don't know. Yeah.
So there's a follow-up question from Krishanu Duda about the DRP1 knockout mice. And they ask, do these knockout mice show bradycardia?
No, if anything, they show more like what I call delay after depolarization, early after depolarization due to the calcium oscillation, like the catecholamine CPVT condition. So if anything, it show I would say arrhythmia in terms of more ectopic beat and less in terms of decrease of heart rate.
Okay, so there is a little bit different direction question, but from Dr. Bridget Nieto, so the heartbeat is different in mouse and the human. And are there any species difference in the mitochondrial calcium dynamic profile or set up the calcium 100 protein?
The short answer is of course, yes. Yeah. The long answer, yeah, we all study mice and rat that beat 300 to 500 times. So from the excitation bioenergetic coupling, it could be really different from, say, human that beats 70 times. Okay, I think the key experiment that is still lacking as of today to address, I may not address your question very directly, but let me try. Okay, the most, I think I shouldn't exaggerate. One of the key experiment that is still missing is because of lacking or a small molecule that is membrane permeable to, say, adult cardiomyocyte, like human or rats, okay? So if we have that compound, then you add that compounds to, say, human heart, and if human heart still beating, yet that compound, if we know block the MCU, then we have to rule out that, because now we are talking acute, not like mice that knock in, knock out. People start to worry about adaptation for the good reasons, compensatory. Okay, so that experiment is still missing, even though there's some indication from RU360, routine rat, but then you always argue how selective they are in terms of blocking the MCU in the intact cardiomyocyte or heart. Yeah. I hope I answer your question. Yeah.
Yeah, I think as I understand that this is very still challenging because we need to generate the new tool for acute inhibition or activation of this. Yeah. I think we have one more question, I think.
I think there's a few more still, actually. There's one from Ron Lynch, who was wondering what your thoughts were on the role of DRP1 and could it be involved in the linkage of the mitochondria into a network? And he references Balabin et al., where they proposed this.
Yeah, and that's why we bring in the we think it's mostly probably cytoskeleton. DLP1 is just that get recruited by the cytoskeleton. That's why we focus on the actin. We think that actin and actin binding protein are the scaffold and may play a role in terms of tethering the SR and the mitochondria. But somehow, maybe there's a binding site or DLP1 with actin. We don't know. We are trying to look into that. And as such, then DLP1 go along with it. Okay. And then the idea of the actin and the DLP1 in the ER mitochondrial junction has been shown by people like Henry Hicks at the and his postdoc now in the Mitochondrial Raj that we collaborating, and that's why we get into the actin. So that idea in a way is not new, but go a little bit, but the time is getting short. And actually, we think that also play an important role in terms of calcium transferring between the SR and the mitochondria through this kind of actin tethering of the two organelles. Yes.
Okay, great. Thank you so much, Dr. Hugh. Unfortunately, we have a time limitation, so we couldn't catch up all the questions. But I really appreciate that everybody's participation for this presentation, and we would like to congratulate Dr. Session Hsu for this year the Davson Award, and we would like to plan to do a similar webinar in next year, 2025 Davson Award. So please come back to this webinar next year. And I would like to thank everybody for participating in this presentation. Thank you very much and have a wonderful day.
Thank you. Yeah, come to visit us or email to me.
Thank you.
I think we can go a little over time if you want to answer the last few questions that are
okay. Shall
here.
we? Okay.
Yeah,
then,
if there's some other
go.
question, I'm happy to.
Yeah, there's a few more questions.
Yeah,
so,
yeah. I will make sure Jim Uchi will buy me lunch next time he sees me.
Okay, that'll be great.
It's overtime. Yeah.
Yes. Yeah, there's a few questions we can go over. Yes.
So one more, sort of changing directions from the scientific aspects from May Helms. She asks, what is the greatest question or challenge in studying ROS and calcium signaling what general advice besides waking up at 4 a.m. and surrounding yourself with smart people would you give to trainees?
Well, first of all, I learned a lot more from training than I kind of teach them. Every training come to my labs, I would look into their eyes and say, do you play squash? And if they say no, I say, I don't think this lab fits you. No, that's just a joke. Yeah. I think in terms of big picture of calcium and ROS, as I indicate in the slide, and I think Doc Wallace had this insight, like integrate so many different diseases that converge into calcium ROS and energetics. And if we think more about this, maybe we can help in terms of etiology of cancer, which now people think is to do with the energetics, okay? The substrates and so on. So it's about the switch between the glycolysis and the oxidative phosphorylation, which regulated by the calcium and ROS. So from that point of view, elucidated this cross-talking, which I only touch a little bit, and I'm still learning that, will help in terms of understand the human health and diseases. So last, yes, as I say, waking up 4 o'clock is not because solely passion for that. It's I'm just not as smart. I need to work harder. Yes, but thanks for the question. Yeah.
Is any? Okay, then last question. So I would like to combine a couple of other remaining questions that is kind of similar. So there is a question from the audience that how the calcium and ROS, this mechanism is important for the non-excitable cell or the cancer, which is usually shut down the mitochondrial function. So is there any like a suggestion for the non-excitable cell research?
Yeah. I only can hand waving because I don't work on non-excitable cell. But having said that, my collaborator, Ethan Yu, the late Ethan Yu, work on non-excitable cells, such as the liver cell. And he studied the diabetes. I use that example, and hopefully I can answer your question. He studied diabetes, and he find the high glucose did 2 or 3 things, which are what I was talking about. One, it causes mitochondrial fission. 2, it enhances ROS generation. What he did not measure is the calcium, which later on we try to measure, and high glucose enhances mitochondrial calcium uptake. So I tend to think, yes, similar principle can apply to the non-excitable cell, but maybe with the catch is because the excitable cell, like high neuron, they fire such a high rate, neuron actually even higher than the heart. So their calcium transport mechanism could be very different. And that come to the point about the time and the calcium concentration, the kinetics and the affinity and on and off rate. So they could have different calcium transport mechanisms because of that condition. Yes.
Great, thank you so much for, I think it's mostly we cover the question that we received from audience. Thank you very much, Dr. Xu. And yeah, a little bit over time. So we would like to close this session and please congratulations for Dr. Xu.
And Dr. Eaton and Dr. Uchi, if there's some question in the chat, please send to me because I may not have, and then I can address them individually. Is there a possibility to collect all the questions
Yes,
in the
sir.
chat or in the question and then send to me and I can reply to them? Whatever, if that is possible. But
That would be wonderful. Thank you.
or they can write to me and ask me, and I will reply to them individually through the email. Okay.
Great. Thank you so much.
Thank
So-
you. Dr.
Okay.
Lee.
Thank you so much. And thank you for all you, and thank you the APS staff and Patricia particularly. I really appreciate it. And we get this through, and hopefully it was all right. Yeah.
Great. Thank you very much, and we would like to close the session. So please come back in 2025 APS Sales Section Davson Hours webinar again. Thank you.
Thank you. So I can leave now, right?